Welcome. I'm John Folk, a lead biologist at ATCC, and today I'm going to present the poster, Newly Established CAR-T Target Antigen Luciferase Reporter Cell Lines Facilitate CAR-T Development. And this, before you on the screen, you can see the full poster, of which we'll be talking about more in the next several slides. Chimeric antigen receptor T cells have displayed remarkable efficacy at treating malignant cancers, particularly liquid tumors. Evaluation of biofunction of CAR T cells in vitro often involves a series of labor-intensive co-culture experiments and immunoassays. In this study, we present the generation of a panel of luciferase reporter tumor cell lines that can be utilized to examine the function of CAR T cells. The panel of selected human tumor cell lines naturally have high expression of CAR-T target antigens on the cell surface, such as CD19, CD20, and HER2. We have completed 5 luciferase-expressing CAR-T reporter cell lines that have been functionally validated to provide a simple, robust, and highly sensitive means to measure CAR-T activity. Well-characterized CAR-T antigen target antigen luciferase reporter cell lines from ATCC provide excellent tools for studying CAR-T biofunction and validating new CAR-T agents for cancer immunotherapy. 5 ATCC luciferase expressing cell lines were generated. Wil2S, LUC2, and Raji LUC2 naturally express high levels of CD19, which can be targeted by CD19 CAR-T cells. Dowdy LUC2 and Faraj LUC2 naturally express high levels of CD20 and can be targeted by CD20 CAR-T cells. BT474 naturally expresses high levels of HER2 and can be targeted by HER2 CAR-T cells. A lentiviral plasmid was virally transduced into ATCC authenticated tumor cell lines. The cultures were selectively isolated using blastocidin. Single-cell cloning was used to generate a homogeneous culture, which was expanded and validated for luciferase expression. The clones were characterized using cell morphology and growth kinetics. Luciferase catalyzes the reaction of luciferin with oxygen to produce oxidized luciferin and light. The light signal can be read by using a luminometer or a CD camera in either a lytic or live assay. Morphology of luciferase expressing cell lines was found to be similar to the parental cell line as detected by bright field microscopy. The growth curve of luciferase expressing cell lines is similar to the parental cell line. The luciferase activity shows a linear correlation between bioluminescence intensity and cell number. In this experiment, 80, 500, 2,000, and 10,000 cells were analyzed for luciferase activity. The luciferase expression was found to be stable for over 30 population doublings. The antigen expression was also found to be stable for over 30 population doublings. Raji LUC2 and Wil2S LUC2 express high levels of CD19. BT474 LUC2 expresses high levels of HER2, and Faraj LUC2 and Dowdy LUC2 express high levels of CD20. HER2 CAR-T cells target and kill HER2 positive BT474 LUC2 as shown in red, and this was detected using the Excelligent system. The blue line shows BT474 LUC2 cells grown in the absence of CAR-T cells. In this figure on the left, BT474 LUC2 can be targeted and killed by HER2 CAR-T cells in a dose-dependent manner. Raji LUC2 and Wil2S LUC2 can each be specifically targeted by CD19 CAR-T cells in a dose-dependent manner, as shown in red. And this can be compared to the non-specific killing of mock CAR-T cells, which were obtained from the same donor, as shown in gray. After 24 hours of co-culture with CD19 CAR T cells, Raji Luke-2 cells, which are shown in green, show a decrease in fluorescent cells, shown in the lower right, as compared to 6 hours, shown in the upper right. In a co-culture experiment with mock CAR T cells, numerous Raji Luke-2 cells remain present after 24 hours, as shown in the lower left panel. Two stained Raji Luke-2 cells, shown in green, from the co-culture experiment previously mentioned, were tracked for 6 hours. The cells were surrounded by CAR T cells, resulting in a decrease of fluorescence when treated with CD19 CAR T cells, as shown in the lower panel, as compared to co-cultures with mock CAR T cells, shown in the upper panel. And we have generated 2 movies where we tracked two stained cells for 6 hours. In the mock CAR T movie on the right, you will see that even though the green Raji Luke-2 cells are surrounded by CAR T cells, you do not see a decrease in fluorescence. On the movie on the right, you see CD19 CAR T cells surrounding and killing Raji Luke-2 cells, as indicated by decrease in the green fluorescent signal. Our results demonstrate the ATCC luciferase reporter system is a relatively simple, robust, and highly sensitive means to study biological processes. They've been characterized and authenticated using cell morphology, growth kinetics, and STR analysis. They have been extensively validated for stable expression of high bioluminescent signals and provide great convenience in measuring target cell killing in CAR T functional evaluation. The well-characterized CAR T target antigen luciferase reporter cell lines from ATCC provide excellent tools for studying CAR T biofunction and validating new CAR T agents for cancer immunotherapy. For more information, please go to www.atcc.org/immuno-oncology. Thank you.